So this information is taken from Thermo Fisher. In the cloning workflow, our first step is to generate the fragment of interest. So this is where we generate a fragment of interest from DNA or RNA or synthetic fragments. To do this, we need PCR reagents. So we need a PCR master mix. So if we're looking at a master mix, we need DNA polymerases, which come with buffers enabling primer annealing. The buffers allow the polymerases to bind and amplify our gene of interest using the primer at a particular temperature. What is a PCR master mix? So a PCR master mix is a premixed solution that contains a thermostable DNA polymerase. It contains deoxynucleotide triphosphates, dNTPs. It also contains a buffer and other additives. The optimized master mixes helps ensure consistent and reliable results and saves time and reduces the potential for errors during reaction setup. So in this regard, you may hear terms like hot start PCR. So hot start PCR is a type of PCR where DNA polymerase activity is inhibited until the initial denaturation step. This inhibition is achieved through various methods, such as antibodies, affibodies, or chemical modifications of the polymerase. The benefits of hot start PCR include increased specificity and yield. Then you undoubtedly would have heard of Taq DNA polymerases. These can be native or recombinant enzymes. So native DNA polymerases are naturally occurring enzymes found in various organisms such as bacteria and archaea. Taq DNA polymerase is one of the best known thermostable DNA polymerases used in PCR amplification of DNA targets. The native enzyme is purified from Thermus aquaticus, YT1. The native Taq DNA polymerase is often preferred for amplification of bacterial DNA sequences homologous to E. coli sequences. The recombinant enzyme is a cloned version of native Taq polymerases with improved properties and functionalities. Another reagent you may need in generating your fragment to introduce for cloning is reverse transcriptase. So this will be needed if you're doing reverse transcription. Reverse transcriptase synthesizes complementary DNA from single-stranded RNA and serves as a template for PCR amplification. Reverse transcription is the process of transcribing RNA molecules into complementary DNA. This cDNA, as it's called, is mediated by enzymes called reverse transcriptase, or RTs for short. Maloney murine leukemia virus, M-MLV, reverse transcriptase, is one of the reverse transcriptases used in molecular biology workflows. However, novel engineered RTs are often preferred for enhanced efficiency, improved thermostability, and higher cDNA yields. How do reverse transcriptases work? Reverse transcription reactions involve multiple steps to synthesize complementary DNA, cDNA. Reverse transcriptases exhibit several distinct biochemical activities, predominantly RNA-dependent DNA polymerase activity and RNase H activity. Firstly, reverse transcriptase binds to an RNA template in the presence of an annealed primer. Next, the RNA-dependent DNA polymerase activity of the enzyme synthesizes cDNA by incorporating dNTPs from the reaction mix. This generates a cDNA-RNA hybrid and is referred to as first-strand cDNA synthesis. If reverse transcriptase exhibits RNase H activity, as in wild-type MMLV-RT, the RNA of the cDNA-RNA hybrid is cleaved during first-strand synthesis. In engineered enzymes, RNase H activity is often reduced to prevent premature RNA degradation. So if you have reverse transcriptases, you've got PCR reagents, you would need oligonucleotides. So DNA oligos are there to act as primers for the amplification of the gene target that you are interested in. So usually when you're ordering these, you need to consider what they call a scale. So that's like an amount, how much you want. And then you can think about modifications. If you are doing, say, quantitative PCR, then there are modifications you can do to allow you to quantify the RNA or DNA as it's being amplified. So for your oligos, you usually order and you're charged based on the length of it. So you can do like 5 to 40 base pairs, or if you're going up to 100 base pairs, you may get a better price. And then they will ask you about the synthesis scale. So this is to do with whether you want 25 nanomole, 50 nanomole, 100 nanomole, or even go right up to the micromole amounts. So in this, when you're doing these kind of molecular cloning, you have to become familiar with the idea of nucleotide synthesis. There are companies that will synthesize dNTPs or NTPs and other modified nucleotides that you can use in your assay. When you're ordering them, you'll be asked about the purity. So high purity, according to high performance liquid chromatography, HPLC, would give you the highest grade and you will pay accordingly. But then there's some that are like desalted and other ways of cleaning up the nucleotides for your applications, which may be cheaper. So you have to become familiar with terms like deoxyribonucleotide triphosphates. You would have encountered them already, but here when you're ordering, you have to have a grasp of what you're actually getting. So deoxyribonucleotides, dNTPs, you would be a mix of DATP, DCTP, DGTP, DTTP, and DUTP. So let's look at the proper cloning part. So the overview here, the kind of things we're going to need are like restriction enzymes, modifying enzymes, DNA vectors, the cloning strategies that are available. So there's one called topo PCR cloning, there's gateway cloning technology, there is Gibson assembly cloning, there's gene art seamless cloning. So where would modern day molecular biology research be without restriction enzymes? These workhorses of the lab have been behind many of the advances in basic biology research and commercial applications for over 40 years. Restriction enzymes, also known as restriction endonucleases, were first identified in bacteria but have been subsequently found in some archaea. In general, restriction enzymes cleave double-stranded DNA. Each restriction enzyme recognizes specific DNA sequences, and cleavage can occur within the recognition sequence or some distance away, depending on the enzyme. The recognition sequences are generally 4 to 8 base pairs in length, and cleavage can produce sticky ends. So you get 5' or 3' protruding ends, or blunt ends. So as we speak, there are about 40,000 restriction enzymes that have been characterized. Wow! And over 600 of those are commercially available. So in the early 1950s, a number of research teams observed differences in the efficiency of bacteriophage infection on different bacterial host strains of the same species. This was described by Grasso and Pagan when phage lambda propagated in one strain of bacteria was used, this was an E. coli bacteria, was used to infect another strain of the same species of bacteria, for example, E. coli K, a marked decrease in the rate of infection was noted compared to reinfection of the host strain, the E. coli C. The new host, E. coli K, seemed to select against or restrict the incoming phage. The researchers also noted that this was not a hereditary phenomenon because the phage that did grow on the new strain could infect that strain at more typical rates after one round of infection. The observed phenomenon was defined as host control variation and became an area of intense research to discover the underlying mechanisms. It was not until the 1960s that mechanisms underlying host control variation were determined to involve enzymatic cleavage of the phage DNA, which led to the discovery and isolation of restriction enzymes. In the early 1960s, Werner Aber observed that the host range determinant resided on the phage DNA, and subsequent experiments showed that methionine was involved in host protection. These findings ultimately led to the proposal of a restriction modification system in which a restriction enzyme and a methylase from the host work together to cleave foreign, viral, or non-methylated DNA while keeping the host DNA protected through methylation. Interestingly, most of the work on restriction modification systems was on type 1 and type 2 groups of restriction enzymes, classified based on aspects of their structure and function. However, the complete utility of restriction enzymes did not become apparent until Kent Wilcox and Hamilton Smith discovered HIND2, the first restriction enzyme of the type 2 class. HIND2 recognizes a specific symmetrical DNA sequence and cleaves in a defined manner within that recognition sequence. This feature, found in most early type 2 restriction enzymes, led Kathleen Dana and Daniel Nathans to use HIND2 in the physical mapping of simian virus 40 DNA, a process known as restriction enzyme mapping. For their pioneering work with restriction enzymes, Daniel Nathans, Hamilton Smith, and Werner Aber were awarded the 1978 Nobel Prize in Physiology or Medicine, with the discovery of DNA ligase in combination with the growing family of site-specific cutting restriction enzymes. Recombinant DNA technology was born. So let's look at the naming convention used for restriction enzymes. The naming convention takes into account three characteristics of the enzyme's organismal origin. So the genus, the species, and strain or serotype to develop a shortened name followed by Roman numerals to represent multiple restriction enzymes from the same strain. So for example, the enzyme HIND3 or HIND3 Roman numerals in earlier nomenclature represents H for Haemophilus, IN for influenza, D for serotype D, and 3 to distinguish from other restriction enzymes from Haemophilus influenza, serotype D. So these classifications have their basis in how the enzyme actually works. So restriction enzymes are categorized into 4 classes based on their structural complexity, recognition sequence, cleavage site, and cofactor requirement. So the type 1s are multi-subunit proteins with both restriction and methylation activity. They require ATP and cleavage site at a variable distance from the recognition site. Type 2s, on the other hand, have specific recognition sequences. Their cleavage site tends to be within or close to recognition sequences. They generate 5' phosphate and 3' hydroxyl termini at cleavage sites, and they require magnesium for their activity. The type 3s have 2-part recognition sequences in reverse orientation. They cleave at a specific distance away from one of the recognition sites. They require ATP. Type 4s have cleavage of only methylated DNA. So type 4s cleave only methylated DNA. Cleavage site is approximately 30 base pairs away from recognition. Because of the specific characteristics of type 2 restriction enzymes, they have become the most commonly used in many research applications such as cloning and forensic DNA analysis. Due to their usefulness in molecular biology research, the type 2 restriction enzymes are the most studied class of enzymes and comprise the largest group, with over 3,500 in this group. So some key considerations when you're using restriction enzymes. By convention, 1 unit of restriction enzyme cleaves 1 microgram of a defined substrate. For example, plasmid PUC19. It cleaves it to completion in 1 hour in 50 microliters under optimal conditions. While the unit definition provides a form of measurement, it should be noted that various DNA substrates in the presence of the same amount of restriction enzyme might have different optimal requirements based on the frequency of the recognition sequences in the DNA substrate and the types of substrate used. In practice, the enzyme manufacturers will often recommend a 5 to 10-fold excess of enzyme or 1 microliter of enzyme per digestion reaction. This is really due to potential variations in quality, quantity, and the nature of the DNA samples. So typically, these restriction enzymes should be stored and used before their expiration date. They should be kept at -20 in a non-frost-free freezer. Restriction enzymes are often supplied in 50% glycerol, and this is to prevent freezing at -20. However, the viscosity of glycerol may make pipetting and dispensing small volumes of enzyme during reaction setup difficult. So it's best to add enzymes last to complete the final volume and to mix well upon addition. Flicking to gently mix the reaction tube content and then briefly spinning the tube will help to disperse the enzyme throughout the reaction mixture and get the reaction solution collected at the bottom of the tube. There's a terminology to be familiar with, and this is star activity. So this is a relaxed activity when the restriction enzyme, under non-optimal conditions, act on recognition sequences with minor differences from their canonical recognition sites. There may be the issue of incomplete digestion. So incomplete digestion is a frequently encountered issue when using restriction endonucleases. Incomplete digestion may occur when too much or too little enzyme is used. The presence of contaminants in the DNA sample may also inhibit the enzymes, and this will result in incomplete digestion. Suboptimal reaction conditions such as buffer composition, incubation time, and reaction temperature are also common causes of incomplete digestion. So some restriction enzymes require cofactors for full activity. For instance, ESP31 used to be called BSMB1 requires DTT, while ECO571 used to be called ACOOL needs S-adenosine methionine. So some restriction enzymes such as ARO and BEVL require 2 copies of the recognition site for efficient cleavage. So for these restriction enzymes, an oligonucleotide with the recognition site is often added to the reaction to enhance enzymatic activity. I think that's enough on restriction enzymes. What do you think? So now let's move on to the cloning part. Be strong.